Assalamu alaikum. My name is Dr. Syed Wakar Shah Kazmi and you are watching IJS Biology. My topic of today is nerve impulse. Before starting nerve impulse, I would like to tell you some important terminologies which is the part of this lecture. And the first one is stimulus. Stimulus is any change in the external and the internal environment of an organism. Stimuli may be the light, maybe a chemical stimuli, or maybe a sound. So these all are called stimulus. And the reaction of an organism against these stimulus or against these stimuli are called as response or responses. The cells in the body of the living organisms which has the capability to receive stimuli are called receptors, while the organs cells which has the capability to produce response are called as effector. Beside it, the electric potential is the capacity to do some electrical work. So these are the terminologies which I use in this topic. I start with the definition of the nerve impulse. So what the nerve impulse? Nerve impulse is information or a signal, a signal about a stimulus. Information or a signal about a stimulus transmitted from receptor to the CNS and from the CNS to the effector. So the information about a stimulus transmitted from receptor to the CNS and from CNS to the effector. So if you define the nerve impulse technically in the technical terms, so we say that it is an electrochemical change. It is an electrochemical change that travel along the length of a neuron from one end to another. So technically we define it as it is an electrochemical change that travel along the length of a neuron from one end to another end. So how generation and the transmission of the nerve impulse occurs? So before studying the process, I would like to tell you something about electric potential. What is electric potential? It is the capacity to do some electrical work. So the work done during the electrochemical change can be called as electric potential. There are two types of electric potential as soon as concerned with the transmission and generation of the nerve impulse. The first one is called as resting membrane potential, RMP, and the second one is called acting membrane potential or AMP. In this video, we will cover the RMP, that is resting membrane potential, and in my next video I will discuss with you the AMP, the action membrane potential. So starting with the resting membrane potential. So how the resting membrane potential is characterized by? The resting membrane potential is characterized by more positive outside, is characterized by more positive outside and more negative inside. It is characterized by more positive outside and more negative inside. So what we say, this state is called as polarized state because we have a positive pole outside and we have a negative pole inside. So this state is called as polarized state. The state is polarized state. Now there is no movement of charges during the RMP. So what we will say, we shall say that the neuron is supposed to be at rest and that's why it is called as resting membrane potential. It's called as resting membrane potential. Generally, we measure the potential from inside as compared to outside. We compare the inside potential from the outside. In RMP, the resting potential is about minus 70 MV or 0.07 minus 0.07 V. And this negative sign, negative is related to outside. This negative is related to outside. It's called RMP as membrane is not being stimulated, or conducting an impulse. Means the inner negative and outer positive shows that membrane is not being stimulated or conducting an impulse. It means is it at rest? That's why the potential minus 70 MV is called as resting membrane potential. Now, how the resting membrane potential occurs along the neuron, I would like to tell you something about the distribution of and the active movement of sodium and potassium ion. Concentration of potassium is 30 times. Its concentration of potassium is K plus is 30 times more, 30 times greater in fluid outside as compared to inside. So, the concentration of potassium is 30 times greater in the fluid inside, greater than fluid inside as compared to outside. 30 times greater inside as compared to outside. That's why the concentration of potassium is 30 times more inside as compared to outside. On the other hand, the sodium ions are 10 times more outside as compared to inside. Repeating this because there's some confusion. Potassium is 30 times more inside the fluid as compared to the outside, while the sodium is 10 times more outside the fluid as compared to inside. So ions are continuously moving against the concentration gradient. Here, the blue color show the movement against the concentration gradient. So this blue color indicate the movement with the expenditure of energy, while here the black color is showing the passive movement and the movement without expenditure of energy. So if we see 3 sodium ion, if 2 potassium ion actively move inside, the 3 sodium ion will actively move outside. So if the 2 potassium ion actively move inside, the 3 sodium ion will actively move outside. Beside it, there is some positive movement of ions through a specialized neurophysiological gates which are called sodium and potassium gate. The question arises, why the outer become more positive and why the inner become more negative? The outer become more positive due to the presence of potassium ion and the sodium ion. These potassium and sodium ion concentration make outside more positive. But the inside, in the presence of potassium ion, why it is negative? It is due to the presence of negatively charged compounds. These are amino acids, proteins, and RNA. In the presence of amino acid, proteins, and RNA, the inner become more negative, while the outer become more positive. And this negative and positive set to poles or membrane is at polarized state. How the leakage of potassium ion occurs? We know that the cell membrane of the neuron has many proteins, and these channel proteins are called potassium gate and the sodium gate. The ions move continuously through these gates. The resting membrane potential, which is maintained until the membrane is until the membrane is disturbed by any threshold stimuli. In a very simple word, this membrane potential retains over the membrane until a threshold stimuli arrive here. And what is threshold stimuli? The stimuli which can change the distribution of ion, that is potassium and the potassium ion and the sodium ion, which has the capability to change the distribution of ions across the neuron membranes. So this is called RMP, or resting membrane potential. I am giving you a very quick review about this whole topic, that what the nerve impulse, the informations or the signals about a stimulus that transmitted from receptor to the CNS and the CNS to the effector. And technically we define it's a wave of electrochemical change that travel along the length of a neuron from one end to another. And electric potential, the capacity to do some electrical work, is called electric potential. There are two types of electric potential. One is called as RMP, resting membrane potential, and the other is called active membrane potential. The resting membrane potential is characterized by the presence of positive ion outside and the negative ion inside. This is called polarized state, and when the membrane is at polarized state, it is supposed to be at rest, and the potential is called as resting potential. If you check the potential from inside as compared to outside, we come to know that it is about minus 70 MV or 0.07 volt minus. So distribution of ion concerned, the potassium ion is 30 times more inside as compared to outside, while the sodium ion are 10 times more outside as compared to outside. The other thing is that if two potassium ion move inside, the three sodium ion move outside. This make outer more positive and inner more negative. Beside it, the inner negative ion is maintained by some compounds which are negatively charged, that is amino acid, proteins, and the RNA. And as soon as concerned with the leakage of the ions, we know there are two passages. One is active passage, which require energy, and the other is the passive one, which follow the gradient, and the movement of ion occurs through passively as well as actively. But movement of ion occurs when the threshold stimuli hit the neuron, and threshold stimuli is a type of stimuli which bring change in the distribution of ion along the neurons. This is all about, and I hope you understand. If you have any question, you can ask in your feedback. Next time, inshallah, I'll discuss with you people the AMP, the action membrane potential. Until thank you very much. Assalamu alaikum wa rahmatullahi wa barakatuh.